This is the second session on the process of transcription. And in our last session, we stopped at the process of transcription, basically the components involved in transcription in prokaryotes. Now we'll move further and we will learn how the process of transcription takes place in prokaryotes. Let me just demagnify this image and let's get back to our original condition here. Right. So we had just stopped here. We had understood what were the different components involved in the process of transcription or requirements for transcription in prokaryotes. We require a RNA polymerase, a multifunctional RNA polymerase, some proteins known as sigma and rho factors. We also need a transcription unit, which is nothing but a gene on the DNA segment, and the transcription unit is made up of three different parts: the promoter region, the coding region, and the terminator region. Apart from that, we require ribonucleoside triphosphates, RNTPs. Now, proceeding further, out of these, let us look at this particular DNA. So let us say we have a strand of DNA here, and we've got these bases here, and let us say we've got, this is the 5' end, this is the 3' end, and let's say this is the 5' end, the 3' end. Now, which of these strands, will this strand or will this strand, which of the two strands act as a template for the synthesis of RNA? Which of the two can act as the template? See, if the direction of RNA polymerase, the direction of synthesis or the orientation of RNA polymerase is again from 5' to 3'. Now, just revising it, the ribonucleoside triphosphates are also added to the 3' hydroxyl groups of the ribose sugar. Right? So the 3'. So all the RNTPs can only be added to the 3' hydroxyl group. This means that the chain grows from 5' end to the 3' end. If something is growing from the 5' end to the 3' end, then in this particular diagram, let's see. What is the possibility of this strand acting as a template, the upper strand acting as a template? The upper strand can also act as a template. Let us just separate the two strands. So let us separate them apart. This is the 5' end, this is the 3' end. Let us say we are only talking about the upper strand. Can this act as a template? Certainly it can act as a template if the RNA polymerase starts working from this direction, right? So this is the 3' end. This will make a complementary 5' kind of a primer here, right? And then this is the 3' end, the chain will grow in this particular direction, right? So the upper strand can act as a template for RNA synthesis. However, if you look closely, if you are a little meticulous in your observation, then even the lower strand can do the same. Because here the orientation of lower strand is 3', 5'. Even the lower strand can act as a template given the RNA polymerase starts from this end. And the RNA polymerase will add a DNTP here on the 3' end, and the next here, and the next here, and the next here. So basically this is the 5' end of the new RNA chain, and this is growing into the 3' direction. So both the strands can act as template, but they don't do so. They do not do so. If both of them start acting as a template, they'll make two single-stranded mRNA molecules which will be complementary to each other, and they will connect together and form a double-stranded structure, and double-stranded structure cannot form a protein. Number one. This is one reason why both of them cannot act as a template. Right. The other reason is why they cannot act as a template. The other reason is that the upper strand and the lower strand, if they synthesize, let us just put a little thing. Let's put A, T, G, C, A, and let's put T, A, C, G, and T. Now let us generate an RNA strand from the upper the upper strand, right? If you generate an RNA strand from there, the RNA strand will be A will be U, right? T will be A, and then G will be C, C will be G, and A will be U. So this is the RNA strand which will be generated from the upper one. From the lower one, what will be generated? we will have A, we'll have U, we'll have G, we'll have C, and we'll have another A here. So you see, two different types of RNAs are synthesized, and both of these RNAs will synthesize two different types of proteins. And for this particular reason also, both the strands cannot act as template. So now the problem is which particular strand is used as the template. Which one? How is it decided? How does the cell decide which particular strand will be used, will become the template strand, which strand will become the non-template strand? Well, this entirely depends on the position of the promoter. So let us say we have the DNA segment here, and I'm going to use a blue for, and let us say we have the promoter region on this side, and let us say we have the terminator region on this side. And let us call it, let us look at the strand, let us just also name it, let us call it the 5', 3', and let's call it the 3', 5'. In this particular case, we'll have to change this orientation. This is not the exact orientation. So if the promoter is present here, okay, we'll just give me a moment. So let's say this is the 3', and this is the 5', and this is the 5', and this is the 3'. And here, if we have the promoter here on this side, and if we have the terminator on the other side, if let's say this is the promoter, in this case, the upper strand, because the promoter is here, if the promoter is on this side, the upper strand, this particular strand will act as a template, right? Because then this will allow, basically the promoter decides which particular strand acts as a template. If the promoter is here, the upper strand becomes the template for the generation of RNA, or basically gets transcribed. However, if the promoter is present on the other side, let us draw the same kind of strands again. Again, we have got the 3', 5' strand, and you got the 3', 5' here. But now, if we have the promoter here, and if we have the terminator region here, then that particular case, the lower strand will act as the template. So this is something for you to remember, that only one of the two strands will act as a template, and which strand will act as a template completely depends on the location of the promoter. Where is the promoter present? That is all you need to remember for a moment. Now let us get deeper into the process of transcription in prokaryotes. Now transcription in prokaryotes takes place in 3 steps. Right, these 3 steps or 3 different stages. The first step is called the initiation, the second is called elongation, and the third is called the termination. The exact same steps we encountered when we were studying the process of DNA replication. Now what happens in initiation? Now here this is a black and white diagram, so let us just make it a little more colorful, and I'm going to use the red color to show the promoter region. So this, the red is the promoter, and let us use green to show the terminator region, and we cannot actually see it, so let us use a yellow. So let's use a bright yellow, and this is the terminator region, this is the terminator region, and okay, all right, so we have the terminator region here, right? Now what we have here in this particular image, what do we have? We have a promoter region. So this is the promoter region here. Now how does the mRNA synthesizing machine, or the RNA synthesizing machinery, how does the transcription synthesizing machinery identify this spot? So the RNA polymerase, before the RNA polymerase can actually go and start the process of transcription, there is a particular protein called the sigma protein or sigma factor. The sigma factor temporarily combines or cooperates with the RNA polymerase. So let us say we have the RNA polymerase here, and the RNA polymerase will associate temporarily with a protein called the sigma factor. Now the function of the sigma factor is to identify the start site or the promoter region. So the sigma factor is basically scanning the DNA, and when it finds its specific, it cannot bind to all the different types of promoters, but it finds its specific promoter site. So it finds the specific promoter site and sticks, and basically, so the sigma factor is present somewhere in between here. They are temporarily associated as shown in the diagram, and the sigma factor identifies the promoter region and binds the RNA polymerase to this particular site. Now once the binding happens, so this basically, this is the, we are studying initiation at the moment. So the first step is binding. What causes the binding? The sigma factor, which is a protein. And now once the binding has taken place, the RNA polymerase starts its function. It melts the DNA. When we say the melting of the DNA, what we mean? So let us say this is the DNA, this is the double helix. When the RNA polymerase, it goes and binds here, what it does is it melts the DNA, by which we mean is that it separates the two complementary strands. It breaks the bonds between them. So the bonds are broken and a bubble is formed, right? And this bubble is formed like this. And here we have got the RNA polymerase. So what basically forms the bubble? If you remember, if you recall DNA replication, this bubble was formed by the enzyme called helicase, right? And here this particular enzyme, RNA polymerase, is executing the function for helicase. It does not require another enzyme, and that is why we call RNA polymerase as a multifunctional enzyme. So this RNA polymerase has bound to the promoter region with the help of the sigma factor, and now it has opened up split up the DNA strand, and this particular structure that has been formed, it is again a bubble, and in this case we call it the transcription bubble. Transcription bubble. It is the equivalent of the replication bubble in case of DNA replication. Anyways, moving forward. So now the two strands have been exposed. One of them is the sense strand and the other is the antisense strand. The sense strand will not make mRNA, whereas the antisense strand or the template strand will make up the RNA, RNA molecule, mRNA, rRNA or tRNA. So now this sigma factor moves through it. It starts to consume RNTPs. There is a lot of RNTP around in the medium. So these RNTP molecules are used by this RNA polymerase. It combines them together based on the complementarity on the template strand, and it starts producing an RNA molecule. And now the RNA molecule, once the RNA molecule is made, this particular structure, which is the RNA polymerase, it reaches the end. It reaches this particular spot here. This region is known as the terminator region. It reaches the terminator region. Let me just repaint it a bit. So once the RNA polymerase reaches the terminator region, there is another protein which is called the Rho protein. So another protein called the Rho protein, at this particular site, it gets associated with RNA polymerase. Temporarily it gets associated, and this Rho protein can actually identify this site. So the Rho protein identifies this site, and then RNA polymerase is basically taken off the DNA strand. So it is disconnected from the DNA strand. It moves out. The double-stranded DNA again becomes bonded via hydrogen bonding, complementary between the complementary base pairs. And once this dissociation takes place, so we have got this here. We got the RNA polymerase, we got the Rho factor here. Soon after, the Rho factor also will dissociate. Alright, one more important thing that I forgot to mention before is once the process of polymerization starts, once the elongation starts, the second step is the elongation. Once the elongation starts, the sigma factor, which was involved in identification of the promoter region, also dissociates during the process of elongation. So let's just rub in, let's just see what all we have done so far. So we have done initiation, we've studied elongation, and we have also studied termination. Now initiation requires sigma factor. Here RNA polymerase has a dual function. It acts as a helicase molecule. It also acts as a polymerase molecule. And what has to be remembered here, one I would like to point out at this very moment, is that the RNA, only one type of RNA molecule can synthesize all RNA polymerases. Sorry, only one type of RNA polymerase can synthesize all the three different types of rRNAs. Only one type of RNA polymerase can synthesize ribosomal RNA, transfer RNA, or messenger RNA. You don't require different type of RNA polymerases. But this is not the condition in case of transcription in eukaryotes. Anyways, so what we have done so far is the process of transcription in prokaryotes, and I hope this particular aspect of transcription is clear to you. I'll see you in the next video with transcription in eukaryotes.